Okay, if everybody can get settled, we're going to begin the special lecture this evening. And since we've been giving this course for the last 10 years, we've always had a special lecturer. And the idea of the special lecturer is to give you all a sense of an academic story in terms of the development of a therapeutic agent and the accomplishment of a success. And we've had a variety of different people do that, and they've always been really a terrific fun for all of us to hear about the successes of our colleagues. One time we had the opportunity to have a pharmaceutical scientist give this lecture, and his name was Frank Bennett, and this was in 2018, and he was in a company that was at the time called ISIS Pharmaceuticals. That name became a problem, and so they changed it to Ionis Pharmaceuticals. And this was a company that was founded by a pharmaceutical company scientist at GlaxoSmithKline, one of the large pharmaceutical companies in the world. And his name was Stanley Crook. And he had a dream to create an antisense therapy platform. And he pursued this for many decades without much success. Until Frank Bennett and then our guest speaker came along, and they really transformed it and made it possible for this dream that Stanley Cook had 34 years ago when he started ISIS and Ionis Pharmaceuticals to have a really a transformational success. And it was of particular interest to me because when I was going through my neurology training, I thought about what would be the best area for me to go into, and I picked epilepsy because I thought in my lifetime it might be possible to have an impact by helping the creation of new epilepsy medicines. We had good animal models for this, and we had some good ideas about how to create better anti-seizure medications. But I saw many patients with neurodegenerative conditions, particularly ALS, which is similar to the condition that we're going to be talking about this evening. And I thought, gosh, if I could do something to help patients with that, that would really be a fulfilling for my own career. But I also thought, heck, in my lifetime, that is never going to be possible. There just isn't any conceivable way that we could reverse a neurodegenerative condition that led to the death of neurons and ultimately was fatal. But I was wrong about that because I'm still alive, and that goal has been accomplished, and you're going to be hearing about it today. Anyway, Frank Bennett told us really a remarkable story of the development of a medication called Spinraza. And he was the pharmaceutical company scientist really responsible for this. But we learned at that time that there was an intellectual leader behind this story, which of course is our speaker tonight, Dr. Adrian Krainer. And so since that time of 2018, I've been thinking, could we get Adrian to come to this course? And finally, Barb and I talked about it, and we said, well, we'll just ask Adrian. And remarkably, when I sent an email, Adrian agreed. So Adrian, thank you so much for fulfilling our dream of having you here at this course. So there's a lot of information on Adrian that you'll find on the internet, several biographies, because he's gotten so many awards that every time that he gets an award, they feel a compulsion to chronicle his life history. So I was able to read those, and I'm just going to mention a couple of tidbits because his history is really remarkable of what he's accomplished, and of course the awards he's gotten for his accomplishments. So you'll find in his biography that he actually grew up in Uruguay. And he was first generation in Uruguay. His parents were in Romania and were subjected to persecution there, which, as I mentioned to Adrian earlier, is actually my situation as well. My father was persecuted by the Nazis in Austria and emigrated to the United States, and I'm a first-generation American as a result of that. But of course, he, unlike me, learning English, he grew up learning Spanish and also French, but not English. He became interested in science when he was growing up, and he figured the best place to study science was in the United States. And so either you taught yourself English or you found somebody to teach you English so that you could then apply for a scholarship to come to the United States. He landed at Columbia University in New York, where he did his bachelor's degree in biochemistry, I believe. Following that, he was able to undertake a PhD research program at Harvard, ultimately landing in the lab of Tom Maniatis, one of the founders of molecular biology. In Maniatis's lab, he conducted the early research that was fundamental to understanding RNA splicing. And then subsequent to that, he took a position at the Cold Spring Harbor Laboratory in New York, where I guess James Watson at the time was the head of the laboratory, and set up his own independent research program. And over the years at the Cold Spring Harbor, he continued to make fundamental discoveries in RNA splicing. But those fundamental discoveries had many spin-offs, leading to the development of the drug Spinraza for spinal muscular atrophy at Ionis Pharmaceuticals. But also, I became aware, actually, of Adrian's work from another company that he founded called Stoke Therapeutics. My area of interest is in the development of epilepsy medications, as I mentioned, and Stoke Therapeutics is the leading company developing a gene therapy for a rare epileptic encephalopathy called Dravet syndrome. And this is really another remarkable story. I don't know whether you'll have time to talk about that today, but it certainly is an indication of another spin-off of your work that has been absolutely remarkable and potentially transformational. So Adrian has won a lot of awards. He's a member of the National Academy of Medicine and then subsequently was elected as a member of the National Academy of Sciences. So I'd be up here for the rest of the evening talking about all of his awards. So I'm going to stop there and let Adrian tell you his story.
Thank you so much. It's a very kind introduction, and thank you, Mike and Barbara, for inviting me. I've really enjoyed today. I learned quite a bit, and I wish I could stay for the entirety of the course, because I really haven't had formal training in pharmacology, let alone neuropharmacology. But so I always worked on RNA splicing since, including my PhD, and this, by the way, is the spliceosome, a structure we didn't determine, but we now know what the splicing machinery looks like. And but you did remind me of when we started collaborating with Ionis, at that time ISIS Pharmaceuticals in 2004. So, and Stan just didn't want to change the name initially because they'd had that name for 20 years and all the logos and things. But I remember going sometimes to visit them, and there would be a limo waiting at the airport with a sign with my name and then ISIS Pharmaceutical. So fortunately that changed quickly enough. Anyway, so thank you for being here. I'm sorry to keep you from dinner for, I don't know, close to an hour, but I'm glad that some of you have your drinks with you. So here we go. So so here's my disclosures and a brief outline. I'm gonna say very little introductory, just the minimum to be able to understand some mechanistic aspects of splicing that for the mechanism of action of Spinraza. Antisense technology, I'm sure you already heard about, and from Gene yesterday, and maybe others, so very briefly. And then I'll give you the introduction to SMA because I believe that hasn't been discussed, and show you how the whole development took place preclinically, a little bit of clinical trial data, and the impact it's having. So I have some patient videos that I think, if I just showed the videos, you probably would get 99% of the picture, and very, very briefly just mention other ongoing antisense projects. So I got started with RNA splicing, and the whole idea, the existence of exons and introns and RNA splicing goes back to 1977. I was a freshman in college, so I didn't even find out about it for a few years. But it was a very unexpected and transformational discovery by two teams, one at Cold Spring Harbor, Rich Roberts and colleagues, and one at MIT, Phil Sharp and his colleagues. And 16 years later, I'm not sure why it took that long, they received the Nobel Prize for that discovery. I need to say something very briefly about alternative splicing because we have constitutive exons that are always included in the mature mRNA, and then we have alternative exons, and these are just 4 examples of common kinds of alternative splicing. So if the exons are the boxes, after splicing you get the mRNA products indicated there. So red or yellow are alternative exons. And so they can be spliced in at least 2 different ways, as shown here. So an exon can be included or skipped, and that's relevant for SMA. But you can also have mutually exclusive exons or different first exons or last exons, and then combinations of that. So this accounts for much of the proteomic diversity because each gene can give rise to 2 or more isoforms. So my lab and others have studied for many years how is alternative splicing regulated, and more generally, how does the spliceosome know what's an exon and what's an intron? And believe it or not, we still don't completely understand that. We have some general principles that we do understand. So if this is an exon, alternative or constitutive, the splicing machinery has to recognize it either all the time or under a certain set of conditions, but under other conditions, if it can be skipped, so that means that this entire segment is seen as an intron. So this, of course, these are just boxes and lines, it's a representation, but you have a long string of RNA, of nucleotides, so what are the features that indicate what's an exon and what's an intron? So you have the splice sites, which are somewhat conserved sequences at the beginning and the end, and a little bit into the exons, at the beginning and the end of each intron. They can be strong, they can be weak. Then you have other elements that we and others started to describe in the 90s. So these have positive or negative influence on splicing. So these are splicing enhancers or splicing silencers. They can be intronic or they can be exonic. And so the way they function is that they are recognized by RNA binding proteins. So activators recognize enhancer elements, repressors recognize silencer elements, and that's the whole nomenclature is very analogous to transcription factors. So the access to these sequences by RNA binding proteins or splicing factors can also be controlled or modulated by the higher order structure of the RNA if it has intrinsic propensity to fold. And also chromatin features can indirectly influence how splicing occurs, how efficiently a given exon is recognized. And so that's less intuitive. But the reason is that much of splicing happens co-transcriptionally. So as the RNA polymerase is moving along the gene and the transcript emerges, splicing factors, regulatory or basal, start binding, and some of the splicing reaction can start occurring. If you think of a gene with multiple introns, it's going to happen not necessarily in the order of transcription, but obviously some introns can already be spliced before the 3' portion of the gene has been transcribed. And so it turns out that if the polymerase travels fast or travels slowly makes a difference, because it can affect the appearance of elements to which factors can bind, or the kinetics of binding. And so there are certain modulatory aspects of transcription and chromatin structure that can influence alternative splicing. So the cartoon at the bottom shows, for example, how a splicing activator works. That's this blob here binds to an exonic splicing enhancer, and then it engages in protein-protein interactions to strengthen or cooperatively facilitate the binding of a basal splicing factor, for example, to the 3' splice site, or the U1 SNRMP to the 5' splice site. And a repressor binds to a silencer element in the exon or in the intron, and it has a negative influence on recognition of splice sites in the vicinity, whether by masking or by competition for binding with an activator. So these are principles that we and others were studying at the time, just before or going into the time that we started working on SMA, which is around 2000. I think this I can dispatch very quickly because it's been discussed. So antisense oligonucleotides, ASOs for short, are synthetic strands of nucleic acids, similar to RNA and DNA, but with various chemical modifications. When you synthesize these, you program a particular sequence into the machine, and that's going to dictate what its RNA target is and where along that RNA target it's going to bind based on the base pairing complementarity. So if we design these to bind to the primary transcript, the pre-messenger RNA in the nucleus, as long as the ASO can get to the nucleus, and if it binds to the target, it can influence how splicing occurs. It can either inhibit binding of a basal splicing factor, or it can inhibit binding of an activator or a repressor of splicing. And I mentioned, but you already know, that because of the large size and in most cases the negative charge, these molecules don't penetrate the blood-brain barrier, so that's a problem we're going to have to deal with. But we were not concerned with that at the beginning. At the beginning, we were just trying to see, what kind of molecule can we design to fix the splicing defect associated with the disease of interest, and then we would deal with the delivery problems in vivo down the line. So let me introduce spinal muscular atrophy. So it's a neuromuscular disorder of motor neurons. It's more pediatric than adult, so half of cases are type 1 SMA that start presenting symptoms before 6 months of age. And so the hallmark is that there is a progressive degeneration of the alpha motor neurons in the spinal cord and in the lower brainstem. So it is a rare disease, but among rare diseases, it's more common than others, just a little bit behind cystic fibrosis, so about 1 in 10,000 newborns. And that corresponds to a 1 in 50 carrier frequency, because this is an autosomal recessive condition. It is or was the leading genetic cause of infant mortality, however, so more deadly than cystic fibrosis. And so the responsible gene was identified by Judith Melke and her colleagues in 1995. And this was difficult. It was positional cloning, and the reason for the difficulty has to do with a region of chromosome 5 that's duplicated and inverted, which made it very challenging. But so Judith identified the causative gene and termed it survival of motor neuron. It turns out that there are 2 genes, SMN1 and SMN2, but SMN1 is the principal gene that produces the protein that's in unaffected individuals. So SMN protein, still the subject of intensive study. One of its functions is to catalyze the assembly of certain RNA protein particles, and among those are the spliceosomal SNRMPs. And so I wouldn't say that it's a splicing factor, but it's involved in assembly of very important splicing factors. But it also promotes the assembly of other RNPs that function in other processes, like histone 3' mRNA 3' end formation. And it has also been implicated in mRNA transport along axons. And so there's always this argument about which function, because the snRNP assembly is a housekeeping function. This protein is essential in all cell types, not just in motor neurons. But that's a debate that keeps going. And we actually haven't really studied that. Our efforts focus on the SMN2 gene, because patients lack a functional SMN1 gene, but they're alive because of their SMN2 gene, which can express a limiting amount of functional SMN protein. And this is unique to humans. So for instance, mice only have one SMN gene equivalent to SMN1. Another characteristic of SMA is that it can have variable severity. So it's been classified into 4 major types, and there are also some subtypes. By and large, this relates to the number of copies of SMN2. So there's copy number variation. The SMN2 gene produces too little protein, but if you have more copies of it, that's a little bit more protein. Hence, onset is later and severity is lessened. So the 3 major types are shown here. And so type 1 is infantile onset with symptoms presenting before 6 months of age. The patients had very low life expectancy, 1 or 2 years. The natural history study by Richard Finkel and colleagues gave a median event-free survival of 10.5 months. And by event-free, it refers to either death or the need for more than 16 hours a day ventilation. A very defining clinical feature is that they can never achieve the milestone of independent sitting. And 80% of type 1 patients have 2 copies of the SMN2 gene. If you have only 1 copy, that typically led to death in utero or within hours or a few days after birth. So type 2 is later onset. Most patients have 3 copies of SMN2. They have somewhat shortened life expectancy. They're able to sit. A few type 2 patients are able to stand also, but they gradually lose those motor functions because the muscles gradually weaken and atrophy, and they have severe skeletal deformities as well. Type 3 is also later onset. Most patients have additional copies of SMN2. They have close to normal life expectancy, and many of them are ambulatory, but again, they may lose that ability as they age. So the genes are present on chromosome 5 and Q13, and so this is an inverted duplication, and so the genes here closer to the telomere are paralogs of the genes here. So some of these other genes have accumulated mutations and become pseudogenes. SMN2 has a very small number of mutations, only one of them in a coding axon, and that mutation is translationally silent, but as you'll see, it affects splicing. So the basis for copy number variation is the instability of the inverted duplication, and you can also have gene conversion events. So for instance, chromosome 5 could end up with 2 copies of SMN2 instead. I have a, okay, it's on. Maybe I wasn't close enough to the microphone. So the exon structure of the full-length mRNA is shown here, and you can see the different segments of the protein as they are coded by the different exons. And underneath are point mutations that can cause SMA. So these are all loss-of-function mutations. And individually, they're rather uncommon, and altogether, they're even not that common. What's more common is to have partial or complete loss of SMN1, most likely by gene conversion. So exon 7 is the last coding exon. And if we look at the SMN2 gene, unlike SMN1, it produces 2 transcripts. via alternative splicing. So about 10% of the mRNA codes includes exon 7, but 90% of the mRNA leaves it out during splicing. And so the resulting proteins look the same, except that the C-terminus, so the last few amino acids of this isoform, are derived from exon 8. And the protein isoform is very unstable, so it's difficult to detect. So in practice, most of the output of SMN2 goes towards making an mRNA isoform that's translated into an unstable protein, and the rest of it makes the perfectly functional SMN protein. It's indistinguishable from that coded by SMN1. It's just present in too low amounts. So this shows that in more detail. So the SMN1 transcripts, exon 7 is constitutively spliced. Translation terminates here, so you have full-length SMN, close to 100%. SMN2 mostly skips exon 7. You get the SMN delta 7 isoform that's unstable, but when the exon is included, you get the same protein, because the only difference in a coding exon is this C to T transition, and TTC and TTT code for the same amino acid, phenylalanine. But these very important studies in 1999 showed that it's the other changes that occur in the introns or in the 3' UTR exon don't matter as much. If you just take an SMN1 gene or mini gene and you change the C to T, you start seeing exon skipping. And conversely, if you put a C in SMN2, you get much more exon inclusion. So these results were presented just before publication at a workshop at NINDS in 1999, and people in that field realized that they wanted to have some splicing scientists look at this. So 2 or 3 of us were invited to this conference. Unfortunately, I attended, and I left there knowing that I was going to work on SMA, because we were studying exactly this problem at the time. How does the splicing machinery know when an exon is an exon? And in fact, we were studying exon skipping mutations in BRCA1, but they affected about 4 people in the whole world, and so we were studying them for mechanistic reasons. And here we have a case where all the patients carry 1 or multiple copies of SMN2, and so if we can figure out why is this happening, and maybe we can find a way to reverse it, to correct the splicing defect, then in theory you could help all the SMA patients. So I came back to the lab and convinced a postdoc, Luca Cartegni, to start working just on the mechanism. What is it about that C to T change? And so he did very nice work, and later Michelle Hastings, another postdoc, also contributed. And so the exon is 54 nucleotides long, and what we found is that when you have a C there, as in SMN1, it's part of a long motif that we had already defined for a splicing activator called SRSF1. And so we could show that it's actually binding there and promoting exon recognition. But when you have a U instead, there's no binding anymore, because the C is an important part of the recognition motif of this degenerate motif that the RNA binding protein recognizes. There may be another aspect to that. It was first proposed by Jim Manley's lab at Columbia, which is that when you have a U there, it may create a binding site for a splicing repressor, HNRMPA1, whose binding specificity had also been studied by Gideon Dreyfus and others. And so potentially both things may be happening at once. And the exon 7 in SMN1 is recognized by an activator, so the splicing machinery finds it right away. But exon 7 of SMN2 is instead bound by a repressor, which hides it from the spliceosome. So we started playing with ways in which we might change the splicing, and I won't get into the early version of this, but the important part of that work is that we made some unexpected observations, and so we published the work in Nature Biotechnology, I think, and or Nature Structural Biology, or whatever it was called at the time. And then we had a patent on it. And so that came to Frank Bennett's attention. And so then he called me up. We didn't know each other personally. And I think this was at the end of '93. And then we decided that we should start collaborating. And we began to collaborate in 2004 once everything was put in place, all the legal agreements and such. And so this was the strategy that we came up with, and that is, as I showed you, the exon is not recognized well because this activator doesn't like to bind there anymore. But the way an exon is recognized is influenced by repressors as well as activators. So what if we find a repressor that it doesn't have a predominant effect, but is there, and then we hide its binding site with an antisense oligonucleotide. So now, even though the activator still doesn't bind, now the repressor can't bind either, and so maybe the splicing machinery will now, the probability of finding the exon increases, and we should get more exon inclusion. And so we started this work in 2004 when a very talented postdoc joined my lab, Yemen Hua, and that's when we began the collaboration with ICES. And so they had proprietary chemistry for antisense oligos. It's no longer, the patents have expired, but at that time, this was the only source of this chemistry, which turned out to be super important. I mean, we didn't know it at the time. The difference in one methyl group, if we had used another chemistry that was available, would have meant that this would not have worked at all. So we, of course, didn't know that. So there's definitely a lot of serendipity and luck involved in this whole process. So there are three types of modifications here. I don't know, Gene, you probably have talked about this quite a bit, so I will only remind you of it. So MOE, which is 2'O2-methoxyethyl-ribose, is this group. It greatly stabilizes the oligo because, unlike RNA, it doesn't get hydrolyzed. It promotes tighter binding to the target, and it prevents cleavage of the RNA strand by RNase H. If it were DNA-RNA hybrid, then the RNA would get cleaved, and we don't want that because the RNA is a substrate for splicing. The phosphorothioate backbone, so that's the sulfur group here in place of a non-bridging oxygen, results in protein binding, and in effect, it allows the oligo to remain in the body longer. It doesn't get excreted as rapidly. And it also promotes internalization through cell surface receptors by endocytosis. And the last modification is that the Cs are methylated at the 5' position because not all Cs in practice, but in certain sequences like CG, the absence of a methyl group means that the RNA or the oligo is recognized as a foreign nucleic acid by toll-like receptors, and you get an innate immune response. In practice, it's easier to just methylate all the Cs, and it's for ease of synthesis. So this is the chemistry we use, although we did explore others at some point. And then we screened. So Yemin screened about 100 oligonucleotides. Initially, we focused on the exon for simplicity, and I don't know if you can see it, every line here represents one oligo and where it's positioned along the 54 nucleotide exon. The whole sequence is shown here. So if it's green, it means that when we transfected it into cells, we saw more exon inclusion. And so we found 2 clusters of oligos that did that. If it's red, it means that it actually promoted exon skipping. So exon skipping was already being used for Duchenne muscular dystrophy. It was still under development. And so we found those 2. And then there are blue lines that show oligos that don't do anything for whatever reason. But there's a lot of interesting information there. So an oligo like this promotes inclusion, and if you shift it by just one nucleotide, it does exactly the opposite. And that coincides with the footprint for a splicing activator that had already been characterized, TRA2 beta 1. So we could also use these in academic research to footprint binding sites and so on. Then we screened the flanking intronic sequences, and we find oligos that, not surprisingly, cause exon skipping because they mask the 3' splice site or the branch point sequence. But upstream of that, we found effective oligos. And then in the intron 7, we found a whole slew of oligos that did exon inclusion. And one of these is already Spinraza. This was, of course, early, these are the initial hits we were getting, and some optimization because we were doing walks of 5 nucleotides, and then when there was a region of interest, then we did micro walks with single nucleotide increments or steps. So that yielded Spinraza already, and so we published that, this set of oligos in 2008, and we already had a mechanism of action in that paper, which is basically this. The way, so this is Spinraza. It's 18 nucleotides long. It sits across the region immediately following the 5' splice site. And so we did some mutagenesis, and actually this silencer was already identified by another group, Singh et al. They called it ISSN1. So we did some mutagenesis and saw that it was a bipartite element, and using biochemical methods, we determined the repressor molecules that bind there. It's either HNRMPA1 or A2, which are very similar to one another. And so we thought two molecules are binding, but Fred Allen's lab subsequently determined that it's only one molecule, but because it has two RNA recognition motifs, it can bind to both parts, and then the RNA loops a little bit in between. Important thing is, why did we choose this one as the lead as opposed to the others? It isn't just potency. If we did a blast search, the only perfect match in the whole genome to these 18 nucleotides is this region of intron 7 in SMN1 or SMN2. And so that sort of conceptually decreases the potential for off-target effects, because if you start relying one or more mismatches, there will be other targets, but the binding won't be as tight, and so you could deal with that with dose response. And also, just because it binds to another RNA, it's not going to cause cleavage. This chemistry doesn't elicit RNase-H cleavage, and the chances that it may interfere with splicing or translation are not zero, but I don't think they're super high. So when HNRMPA1 binds here, it represses recognition of the exon. When the oligo binds here, we could show biochemically, we measure higher occupancy of the 5' splice site by U1 SNRMP, which is the initiating step in spliceosome assembly. This is the chemical structure of Spinraza. And just kind of this cartoon shows the mechanism a little bit more. If this is SMN1, you have SRSF1 bound there because of the C. If it's a U, it's no longer bound. And HNRMPA1's there, but you can block its binding via Spinraza. And so this worked beautifully in vitro. Now let's start using in vivo models. I'm losing track of time, so let's see. Okay, thanks. So at that time, several labs are engaged in generating mouse strains to study that. Now, if you think about antisense oligonucleotides, so it's a hybridization-specific mechanism, and so the chances that you can go across species on target, it depends. Well, for one thing, mice don't have the SMN2 gene. They only have SMN1, which is constitutively spliced in exon 7. And so, the logical way to proceed is to have a transgenic mouse. And several groups were constructing those, and they weren't all available at the same time, so we had to make some choices. And we went with mice generated originally by Hong Li's group in Taiwan. So he prepared two strains. One is a knockout of the SMN locus. So the homozygous knockout is lethal, the heterozygote is normal. And then he also made mouse that are transgenic for the human SMN2 transgene with 4 copies of the transgene. So we certainly use that strain because it has a normal lifespan and it's fertile. It has a weird phenotype, which is gradual necrosis of the tail and ears. But we did many experiments there because it gives us a way to do, for instance, ICV injections or infusion in adult mice. And then there are more severe models that only live a few days, so you don't get to that point. But it's also, obviously, we wanted to study those as well. If you cross these two strains, then half the mice have 1 copy of the mouse SMN gene and only 2 copies. They're hemizygous for the human transgene. And so these mice are normal because of the wild-type SMN copy. But the other half of the litter are homozygous null for the mouse locus and only 2 copies of the transgene. So you can see that they're smaller. This is at postnatal day 7. And they only live for another 3 days on average. So it's a very severe phenotype that we liken to type 1 SMA. It gives us 10 days to intervene. And so in this case, what we were doing is intracerebroventricular injections into the newborn mice. So at postnatal day 1, we could take a needle. The mouse only weighs 1 gram at that point, but we can inject a dose, for example, 20 micrograms. And so we are injecting into CSF, and so we see what happens to the mice. I mean, we sacrificed them at postnatal day 7, and then we took out the spinal cord, extracted RNA, and analyzed. And so the 3 control mice on the left are injected with saline. The ones on the right were injected with Spinraza dissolved in saline. And I forgot to say, but I mean, I'll come back to this. The chemistry allows internalization without, special formulation. So we're not using lipid nanoparticles or transfection, except in cell culture. In cell culture, the receptors sort of decrease in abundance. So in many cell types, if you want to get oligos into a cell, you have to use lipofectamine or something like that. But this is one of the few things that works. It's easier to do in vivo sometimes than in vitro, than in cell culture. So what you can see is that there's a dramatic switch in splicing from mostly exon skipping, this is RT-PCR, less than 15%, to more than 80%. And that already tells us that all or most of the cells in the spinal cord, so neurons and glia, are taking up the oligo, because let's say if you only achieved 50% delivery to these cells, then you couldn't see this much of a switch. We're looking at bulk RNA here. At the protein level, we are seeing some basal level of protein expressed from the transgene, but in the presence of Spinraza, it goes up substantially in the same time period. And that's enough to make a difference in survival. So this is a Kaplan-Meier. And so we see a 70% increase in survival. But this is just a single injection, and we couldn't inject more than 20, at most 30 micrograms into a tiny mouse. It wouldn't tolerate that, independent of the sequence. That's a class effect. But as the mice get older, they can tolerate much higher doses, so they're bigger in size, and something else is changing too. So we also found other ways to deliver the oligo systemically, because you can reach the CNS before the blood-brain barrier has fully matured, which takes, I think, 5 days in mice. And or we could do repeated injections. So if later we could achieve a 25-fold increase in survival to 250 days instead of 10. So very, very striking. And then the mice weren't just alive. They could run around happily, and we did all kinds of quantitation of motor function. So it was very striking. So at that point, with that type of data, Ionis was quite happy, thanks to Frank Bennett's leadership, to start a clinical development. So in my lab was really only involved in the preclinical stages, and I count that from around 2000, 2001. We continued that through 2011, but including lead optimization and such. And so then Ionis started the other steps that you've learned a lot about these days, including tox studies in non-human primates. The gene duplication that gives rise to SMN1 and SMN2 occurred in primates, but the C to T change is only in humans. So mice have, in effect, 2 copies of SMN1 in each chromosome 5. And so there's nothing to fix. So we can't really look at target engagement in the monkey, but everything else, the dose range that's tolerated, all the tox studies and neurotoxicity and the biodistribution and different ways of delivery, all that was studied in non-human primates. So then Ionis started a phase 1 trial, and then around that time, then they partnered with Biogen, and so Biogen sponsored the phase 2 and phase 3 trials and does a commercialization. So at the end of 2016, the drug reached approval. So we're looking at a 15 or 16-year timeline. And in the clinic, as you probably know, because there was a beautiful lecture this morning about delivery methods, what was decided is to do intrathecal dosing, so by lumbar puncture. And so it depends who you talk to. I've never had a lumbar puncture myself. The clinicians who administered think it's trivial. People who've had one don't think it's trivial because occasionally you get a big headache. But I think the procedure has been optimized. It definitely is very well tolerated, and there were nice studies done for that in these patients. And I haven't really met SMA patients or families who aren't actually looking forward to their next injection because they realize it's helping them, and many of them say, I wish I could have it sooner, or can't you give me more drug? So I don't know that it's addictive or anything like that. But so other methods were considered. So when we started the experiments in adult mice with 4 copies of SMN2, we actually were using microosmotic Alcid pumps, so intracerebroventricular infusion. And the original idea was to use a Medtronic's pump, like one that was shown this morning or this afternoon, I forget. And it turned out that Ionis and Medtronics couldn't really agree on commercial terms. I think Medtronics wanted 50%, and that was out of the question. And so then by the time the NHP studies were being done, Ionis looked at bolus injections, and actually they found better biodistribution than with the pump they were using, which I don't think was a Medtronics pump, but I don't know exactly what it was. So in the end, it was decided that intrathecal bolus injection would be done. Other things that were discussed included an Ommaya reservoir through the skull. The neuromuscular people I talked to, really are appalled by the thought. But when I talk to neurosurgeons for pediatric oncology, they think that's a trivial procedure. Why not? So I think it depends who you talk to. But I mean, this has worked rather well because of the long half-life of Spinraza in the CNS. So in adult mice, we measured a 200-day half-life. Compared to systemic injection in the liver, you have a 20-day or so half-life. So through all the extensive studies that were done in NHP, and then the dose escalation studies in the clinic, the current doses are 12 milligrams, irrespective of the age of the patient or whether it's type 1, 2, 3, or 4. And, but the assumption is that the more severe, type 1 patients may need more drug because they have fewer copies of the gene. And anyway, the loading doses are given 4 in about 2 months, but then maintenance doses are only 3 times a year. And currently, Biogen is investigating higher doses because they never really achieved the maximum tolerated dose in the clinical studies. There was a rush because there was no other treatment to decide on a dose and start getting the label and marketing right away. So studies have been done about how the oligos are getting distributed to the CNS. These are studies in NHPs, and so the parameters that matter are the location and the volume of the administered bolus. And then the drug, there's convectional transport that has to do with the heartbeat and breathing, and then helps propagate the drug through CSF and into the neural tissue parenchyma. So I showed you data for the spinal cord, but the brain results on RNA and protein are exactly the same, at least in mice. And then this is followed by receptor-mediated endocytosis and or micropinocytosis. This is not something we've investigated, but here are some references. And actually, only a small portion of the drug ends up where you want it. So I don't know if this is correct or not. I've heard some skepticism, but 96% of the drug goes to the periphery, but most of it is rapidly eliminated in the urine. And there's a much shorter half-life, so there's only transient exposure in peripheral tissues and organs. The clinical trials started in November of 2011. Altogether, they enrolled a little over 300 SMA patients, and those are infants, children, adolescents. And some of the children and teens have been on drug for over 10 years, if those that were in the initial trials and remain on extension studies or just commercial access. The infant trial started a bit later, so it's been at least 8 years for a few of the patients. Now, the clinical trials showed statistically significant and improvements, and also clinically meaningful improvements in motor function and improvements in survival. And we had no idea at the beginning of these trials what could be expected, because there was no other treatment. And so is it reversible? Can you just hope to stabilize it, but can you expect an improvement? Because if you've lost motor neurons, they're not going to come back. But in fact, one does see continued motor improvements over time. So it's not just preventing the natural decline. There were 2 phase 3 studies. They had kind of a placebo arm using a non-sham, sham injection as a no treatment control. And so this is blinded, randomized, et cetera. I don't know where Shai is. Hello. And but they had a pre-scheduled interim analysis of the data so that allowed them to end the trials a year ahead of schedule, because the results were very clear-cut. And there's another trial, Nurture, which was open-labeled on very young pre-symptomatic patients, gave really remarkable outcomes that I'll show you. But this is the ENDEAR phase 3 study. This involved type 1 SMA infants with either 2 or 3 copies of SMN2. And what you can see is that at the top is event-free survival. So that's death or the need for permanent ventilation. There's a very clear separation between the nusinersen treated versus the sham injection controls. And overall survival is at the bottom. So this is the primary endpoints that convince the FDA that to rapid approval. And I'll show you more data, but I want to run this video, which is a patient named Cameron who participated in the phase 2 open-label study. So he's type 1. What do I do? Okay. All right, so you can see that he does belly breathing and is essentially immobilized. He was diagnosed at 5 weeks of age, given 6 months to live. But at 7 weeks of age, when the video starts, he started the treatment. And so then you'll see how he progresses over time. Here at 4 months, and he's already moving his arms a little bit and his head. 4.5 months, he can reach, he can grab an object. So he's already doing better than expected, and of course he's also aging in the process, and so some of these are developmental milestones, but he might not have reached them otherwise. This is at 9 months. And this is at his first birthday.
You're working out?
Okay, so now he's bouncing, and he's holding his head, not without some difficulty.
Yeah, you're doing such a good job.
Look at that big boy.
Can you let
So
go?
the type 1 patient clinically could never sit independently, but now he's beginning to be able to stand.
Stuff up.
Come and get it.
Now he's 2 years old.
Roll the baby. Roll the baby. Oh, what a strong boy. You're going to go get that train? What is that? Hey.
So at that time, Cameron was using a motorized wheelchair, very typical for SMA patients, but he no longer needs that. He uses a regular wheelchair.
Push yourself.
And then this happened.
Push, push. Give him a little help. Push, push, push. Push, push, push. Yeah, keep going. Push, push, push. He is riding a freaking bicycle. So take that, SMA.
Oh my God.
So Cameron turned 9 last December 31st, and he's doing great. So let me get my laser back. Yeah, so he uses this kind of wheelchair now. And these are old pictures, but he could already sit independently, and including on his dad when they're doing martial arts. So back to some clinical trial results. So this is a motor functional scale, HINE-2, over 1,200 days, and these are 17 patients, like Cameron was one of them. And the natural history of SMA, you never see improvements in motor functions prior to the treatments being available. The maximum motor function was at the time of diagnosis, but there's a gradual, steady improvement, and you could think at this point it might be a placebo effect, so that's when the phase 3 trial came in. Now, 40 of these infants initially weren't getting the drug. They were getting a sham injection, randomized, blinded, and eventually they would get the drug, but not for about a year or so during the duration of the first phase of the study. And 81 infants did get the drug, and you can see these curves superimposed rather well, the open label and the placebo-controlled. Then there's a nurture study with patients were treated upon genetic diagnosis, but before the onset of symptoms, and that's this curve. So the slope is much greater. This approaches normal motor development for infants. There's 15 patients in this study. And so they all had only 2 copies of SMN2. There were, I think, another 5 patients had 3 copies, and then they plateaued faster. So those results were quite remarkable. So what about for type 2 patients? So the way the trials were designed, they had to do different motor function tests, so they needed to follow instructions. They had to be at least 2 years old. So these are 2 different motor functional measures, so Hammersmith scale, but this is from the phase 3 study. The control group is flat, but the treatment group is improving, and the difference is clinically meaningful, also statistically meaningful. And then this is another, so the upper limb module. There's I guess I wasn't seeing this curve, so it's not completely. This curve is flat. This one improves a little bit initially, but then declines. But clearly, there's a difference upon treatment. And I think most importantly, you don't see a plateau. There's a continued improvement. So I'll show you a video from one patient that was in this study.
And the point.
Oh.
That's Emma Larson.
And the point.
Emma, do you want to say hi?
No.
Emma, she is a 3.5-year-old, sassy, smart, fun little girl.
Emma was diagnosed with SMA, spinal muscular atrophy. It's self-explanatory. Muscular atrophy, these muscles are starving for the protein from these motor neurons, and her little body doesn't produce that protein or an adequate amount.
They said that she would never stand, never walk. It would eventually affect her upper body and her breathing. It's very scary enrolling your child into a clinical trial. We don't know what the effects are going to be long term. We still don't, but at this point we knew if we didn't do anything what the outcome would be. She was diagnosed at 1.5 years, and at that point they said that she couldn't enter into phase 3 until she was 2 years old. In that 6-month period, she went through a lot of regression. It got to the point where she couldn't even sit up and hold a bottle without tipping over. Her diagnosis was pretty grim. Sorry. Okay. Her lifespan would be shortened, and basically we'd watch this disease progress over time. On the day of her 2nd birthday, we literally drove to Columbia to get her screened for the Ionis nusinersen trial. Then 3 weeks later, she got her first injection, and we started noticing changes in her around the 2-month mark. She crawled all the way from the living room to our master bedroom, and we were just in shock. She now stands We have little orthotics that help her now with a walker, and we just started her with crutches. Not only that, the disease has stopped progression. She's no longer lost anything. In fact, every time she goes in for another injection, she gets a little stronger.
So this video was made 6 years ago. Emma turned 10 last February. She's doing great. This is also from a few years ago when she was not she had never stood before, but now she could stand independently.
One,
And
two,
seems a little bit reluctant to believe that she can do it.
6, 7, 8, 9, 10, 11, 12. 1, 2,
Anyway,
3,
she counts to about 100, so I shift. I want to show what happens with adult SMA patients. So they have advanced disease, so this is a friend of mine in Germany. He's in his 40s, Phil Harold, and he's a quadriplegic. He could only really move his thumbs, and he needed a ventilator constantly. But he's an artist, so he creates digitally somehow. I don't know how. These paintings and sculptures, and a very creative guy. And so so I met him because they were writing his autobiography, and they had questions about the mechanism of the drug. But anyway, after just a few doses, he said that he didn't feel cold all the time. He started moving fingers that had been paralyzed for 15 years, and he could go a few hours a day without the respirator. And so this is a type 3 patient, already post-approval. She's Sophia in Canada. So she was able to walk, as you see on the left, but with difficulty. Then she gradually lost that ability. She was approximately 2, and then she started taking the drug at age 4, and you can see on the right she was walking much better. And a little later, parents sent these videos, things that she's able to do. I mean, she's walking, obviously not perfectly, but I hope she continued to improve. But one thing that the mother shared with me, a list of things that Sophia was able to do after the treatment, after starting the treatment. And most of these things are things that we take for granted, things that are never measured in a clinical trial, but that make a huge difference in quality of life, in my opinion. So I'll get back the laser pointer. So the drug achieved certain milestones that for the first time. So it was the first approved medicine for SMA, first approved drug that corrects defective splicing. The same year, there was conditional approval for Duchenne exon skipping oligos, but those are actually introducing a splicing defect to bypass frame shifting mutations. So it's kind of the opposite. It's a different concept. It was the first approved nucleic acid therapy for neurological disease. I think that this pre-symptomatic treatment was really quite striking, that you can at least delay, if not prevent, the onset of a neurodegenerative disease if you do the treatment early enough. And it's a disease-modifying therapy for neurodegeneration. And so then it was recognized in 2017 by the Prix Galien in the US. And then later, and I think 6, because these are given in different countries, so it won a bunch more awards. And then this last year, there was a 50-year anniversary of the Galien Prize that had been postponed because of the pandemic. And Spinraza won the silver medal in this category, Best Orphan and Rare Diseases Drug from 1970 to 2020. So I certainly didn't expect that. There were some great contenders. But to me, it's amazing because, when I started working on RNA splicing, I always felt that it's relevant for disease and maybe it can be used for therapies, but I wouldn't have dreamt that it could get this far. So that's really a dream come true. And this video is running, and I want to explain what that is. So this is a patient with type 1 SMA, genetically diagnosed. There's now newborn screening. And he started the treatment at 6 days of age. He's now 16 months in the video, and he's learning to walk like any normal infant. And so that's really remarkable. So that's based, this is not a participant in the nurture trial, but that's basically what was observed in the nurture trial. And some of those patients are already 5 years old, so hopefully those gains will be maintained and continued. So there are more than 13,000 patients that have been treated with nusinersen or Spinraza. There are now, this is a little old now, there are 48 states plus recently the Washington, D.C. Do newborn screening for SMA in all newborns. So it's now up to 99% of newborns in the U.S. And so that means that after diagnosis, you can start this pre-symptomatic treatment. And Biogen is also doing this trial, the VOTE, where they're testing higher doses to see if there could be an additional benefit. And they're considering other chemistries, it's early days still, to see if they may reduce the dosing frequency as well. And since then, 2 other SMA therapies have been approved. One is an AAV gene therapy, and one is a small molecule. So just briefly, there's so far 9 antisense oligos that have been approved, although one was withdrawn a long time ago. 4 of these are for Duchenne muscular dystrophy, so they're targeting different exons, but it's similar principle. Only one so far of these approvals is in the CNS, and that's Spinraza. But I think, well, there are many in the pipeline, so I think there will be more. And of those 9, 5 of those are splice switching oligos. So the Duchenne drugs, these 4, use a different chemistry, PMO, but the principle is to bind to an RNA. In that case, these are blocking splicing activators and thereby promoting a skipping of an exon. So I think this type of oligo with uniform chemistry is highly versatile. We can do many things with it. We can downregulate gene expression, upregulate gene expression, change isoforms. So I think that there's great versatility. And so I want to recognize that Reiser Cole was the first person who showed that an antisense oligo could be used to switch splicing, to change splicing. He did that in a cell-free system initially. So there's different mechanisms of action. In the interest of time, I'm not going to get into any detail, but so we can disrupt normal splicing to downregulate gene expression, and so we're doing this for some projects in the lab. So there's many different ways to downregulate gene expression, for instance, siRNA, but right now we have this chemistry, we know how to deliver it to the CNS, unformulated, it has long half-life, so I think that could have some advantages. We can switch between isoforms, so I think that's pretty unique to this platform. Risdiplam, the small molecule for SMA, could do that, but so far it's only been proven with that one target. I don't know if it's going to be a generalizable approach yet. Stoke Therapeutics has this great candidate in phase 2 studies already for Dravet, as was mentioned. So far, unformulated SOs are very effective in the CNS, in the liver. That's a preferred target if you inject systemically. The eye is also very attractive. They have long duration of action in the CNS and the eye. We've done experiments in mice, and Michelle Hastings' lab has also done where you deliver prenatally in the mouse to the amniotic fluid, and then you could see effects, long-lasting effects postnatally. I don't know if that can be translated to the clinic, but that could open up potentially for developmental disorders. So these are the projects, antisense projects we currently are working on. The ones we did with Ionis are SMA, then familial dysautonomia, which is a very, very rare disease. Right now, it's the oligo we developed is with N-Lorem that is finishing tox studies, and we hope there will be a first-in-human study this year. And we also worked on oncology targets. The one that I'm really excited about currently, but I won't talk about it because it's too late, is downregulating a mutation, a dominant mutation in a histone gene for a pediatric brain cancer. And then I listed here work from others, including the Dravet. But there's maybe 50 to 100 other oligos, including the RNase H mechanism, in the pipeline in industry and academia. And so now I want to acknowledge this a picture of people currently in the lab, or some of them at least. The heroes in the SMA work were Yimin Hua, who did the vast majority of the preclinical work. Also, another postdoc, Ken Sahashi, contributed to that, a technician in the lab, Bingshu Liu. Before that, the mechanistic work was done by Luca Cartegni and Michelle Hastings, and it kind of opened up the potential solution that we then explored. We certainly couldn't have done that without Ionis, especially Frank Bennett and Frank Rigo, but many other people at Ionis as well, and Biogen later on. Clinical trials were done in multiple centers in multiple countries, lots of great work there, and SMA patients and their families obviously were crucial as our sources of support. And the last thing I want to show is this, because I have received many moving emails and letters from patients or their families. I remember when the drug was approved, that same evening I got a chain letter because it was 2 days before Christmas when the approval came, and so anyway, this email was signed by 100 patients or parents from all over the world. I still have that. It's very moving, but this one is my favorite. So this is a parent whose son started taking Spinraza, and he says, "My son Zach is receiving his first dose of Spinraza today. Our deepest gratitude for you and your lab for doing this important work. I pray that you and your lab will continue to be blessed with overabundant funding and brilliant graduate students." And this proved to be true. Thank you.
Questions or comments on this absolutely wonderful and moving lecture? Barb.
That was awesome, Adrian. I'm back here tearing up with some of those videos, being the mother of a special needs child. But my question to you is are these the 3 therapies that are used for SMA? Are they given in, or sometimes multiple therapies given, or how does that work?
They're very expensive, all of them. So, and there haven't been any head-to-head studies. And so then I think clinicians, parents decide, they're possibly influenced by marketing. But there are some patients that took the gene therapy covered, let's say, in a clinical trial, and then Spinraza or risdiplam covered by insurance. So there are patients that have taken more than 2 therapies, and it may be that there are certain benefits to each, and there is a logic behind it, but the high price currently hasn't, so I'm not aware of what there are ongoing studies, which I think are not, I have to be careful what I say. Well, basically each of the companies is doing studies where patients are switching from another drug to theirs, but I'm not sure that that's done for something other than marketing, but useful information may emerge from those. Yeah.
Hey, Andrew, this is a fantastic talk. It's a wonderful story. We all kind of hope in the future we can work your work over there. So actually, I worked on another splicing mutation from the gene I study, and I try to create a disease model for that. So I learned that there's kind of intron definition model to define where's the intron and exon definition model. So I want to ask you some detailed things, but can you just comment on, if I have a mutation affected the splicing, how can I think about this? So if you have more time, I want to ask you offline regarding the mutation I have.
No, I'll be happy to discuss with you. In general, I think there are many mutations that are potentially correctable by this approach. If you have a mutation in the first nucleotide of the intron or the last nucleotide, I think those are maybe impossible because that's engaged in the chemistry. But there may be some new developments that somehow make that possible.
Yeah, it is the last intron, the next one is the exon. So it's very difficult. That's AG.
So it's the last nucleotide of
yeah,
the intron?
It's AG, that G.
Sorry,
It's like the last two is AG, right? AG, then the G
yeah,
is mutated.
yeah. So which nucleotide is mutated? The AG or
the G mutated back to A.
the G? Oh, in the exon or in the intron?
In the intron.
Yeah. That's a tough one, I think.
Okay.
Actually, yeah. Somebody just a parent contacted me about that. Yeah.
Very nice talk, yeah. So
Thanks.
I just have a question about the dosing. I know you mentioned first dose. Maybe I didn't follow with you. So you said the first dose, and do they continue to receive like the dose?
The way it was done, so the studies were, the clinical studies, phase 3, followed, so dose escalation studies, which were 1, 3, 6, and 9 milligrams. There were no adverse events that related to drug dose, and things looked promising, and so they decided to add one more dose. They talked to the FDA, and they added 12 milligrams. And then, then the separation between injections was not the same in the clinical studies versus what was ultimately approved. So there was some discussion there, and well, let's go with the highest dose, and we think type 1 SMA infants probably need high dose, so let's give them the same as even the adults would get. And then the idea was just to have several injections in the first 2 months to kind of build the drug concentration to rapidly. And that was mostly thinking about the type 1 SMA infants who have a rapid decline, rapid motor neuron loss, so you want to correct things quickly. And it's possible that gene therapy might be more effective for that, but this is all theoretical. And then the maintenance doses, that's every 4 months, because that's based on the studies that were done in non-human primates to look at the half-life of the drug and so on. But it's all being revisited, so much higher doses are being tested now in the DEVOTE trial. I think 30, 40 milligrams, I forget. So some other ASO drugs are being tested up to 70 milligrams. And the principal target here is the spinal cord and lower brainstem, and I think it gets to the brain, but you may have to start with higher doses if your targets are deep in the brain. There's a lot of speculation there, I think, but one can go higher, that's for sure.
So unfortunately, I am going to have to cut our formal discussion off short.